Hello, everyone. Tonight, the Minnesota Lyme Association wholeheartedly welcomes Dr. Laurie Radovsky. Dr. Radovsky has been helping and healing people with tick-borne illnesses for close to 25 years. She has also been a mentor to numerous clinicians and is a faculty member for the Vectorwise CME online courses for treating people with persistent Lyme and co-infections. She will continue this work but will be retiring from patient care. We want to thank her for all the years of care and all the people she has helped. If you have questions for Dr. Radowski, please type them in the chat and we'll ask them after her talk. I'm
And,
proud.
and
Yes.
yeah, there'll be plenty of time for questions because I don't have a whole hour's worth of stuff
Oh,
to talk
yeah,
about. So
no
yeah,
problem. Okay.
yeah.
Well, I'm proud to introduce Dr. Laurie Radowski.
And I am so honored to be speaking. And you just said that Minnesota Lyme Association is 20 years old now.
Yeah, in
So
April.
that yeah, so that's kind of the time frame of when I've been treating it. And when I was asked to talk, I really thought what a great opportunity for me just to reflect on, what's changed, what hasn't changed, and to just really have a chance to think about this whole crazy ride that I'm on, but you are as well. Um, and I don't want to just talk about my career. Like you guys would probably be very bored for me just to say, what I've done. But I thought it would be kind of fun to look at more of the like what has happened over this time. I've been experiencing it, but it was really helpful for me to kind of go back and think how things have changed. Um, and I don't want to start even 20 years ago. I actually want to start at the beginning, uh, because it helps to put things in context, and it's also really cool information. So, so I did a little bit of research on Lyme disease itself, and was surprised to find out that it's actually been around for thousands of years. There was a team of researchers at Yale University that analyzed the full genome of Borrelia burgdorferi in North America and figured out that it has been present here for 60,000 years. So way before human beings were uh, on this side of the world. The oldest known infected person was Otzi, the 5,300-year-old mummy found in the European Alps. Any of you heard of this story? Some of you have? Yeah. Um, so Otzi was a hunter and was actually probably murdered in the Italian Alps and mummified so that his body was found and has been analyzed up the wazoo. And he was found to have Borrelia, not Burgdorferi, one of the European strains. There have been cases of what are clearly Lyme disease, both in Europe and the United and North America, as far back as 1764. So this was called by a lot of different names: meningopolyneuritis, Guerin-Boujadou syndrome, Banworth syndrome, Avcelius's disease, and one of the European Borrelia strains is Borrelia avcelia. Montauk is actually at the eastern end of Long Island. So Montauk knee, swollen knee, and sheep tick fever. But it wasn't until, as many of you know, 1975 that what we are currently calling Lyme disease was identified because of an outbreak on what was initially thought to be juvenile rheumatoid arthritis in Lyme, Connecticut. In 1980, Willy Burgdorfer, who was a German scientist, discovered the spirochete Borrelia burgdorferi while he was doing research on Rocky Mountain spotted fever. And he was the one that made the connection between the European strains of Lyme disease and North American ones that said, oh, this is a spirochete, just like in Europe. So the controversy around Lyme disease and the diagnosis and treatment was actually pretty soon after it was identified. While the conventional medical world promoted the idea that Lyme disease was easy to identify and responded to a short course of antibiotics, some clinicians encountered patients for which that was not the case. So some patients did not get better with a short course of antibiotics. Some patients got better but relapsed. Some patients fit the clinical picture of Lyme disease but had negative tests. And testing in general has been a huge issue since the beginning. So the current test that is the official test is the two-tiered test. So the first step is ELISA screening. And if the ELISA is positive, then the Western blot is done. And if the Western blot is positive, then people are considered to be infected. So this is actually not an unusual way to do things. HIV testing is actually done in a two-tiered test. So there's the screening. And if you think about screening is you're trying to make sure you don't miss any cases of the illness. You're going to cast a wide net and you're going to get in that net things besides what you're trying to find. And so the second tier, the Western blot, is designed to like get rid of the porpoises and just stay the tuna in your net. But unfortunately, because scientists wanted to make sure that they didn't overestimate cases, because this testing was actually initially created to for surveillance. So it was to figure out where is Lyme disease. But unfortunately, it became kind of the standard of care, like the same way that you would do strep screening if you have somebody with a sore throat. So the problem is because the testing is very specific, so it doesn't have a lot of false positives. Anytime you have very specific tests, you have very insensitive tests. And so you get a lot of false negatives, which is fine if what you're doing is just trying to figure out the prevalence of an infection. But this was not the case. And so imagine if we had a test for strep throat that was like as inaccurate as missing 50% of the cases. So, the testing that we have is still not much better than it was 20 years ago. I mean, the conventional testing. The other problem is that the testing was not really created to be an in the moment test for acute illness like strep throat. Really, it can take up to 6 weeks before a test converts to positive, which the CDC itself says that this is not a diagnostic test. And testing is actually not the only issue. The entire approach to treating Lyme disease is based on only 8 randomized controlled trials of American patients with EM rashes and only 4 studies evaluating retreatment of patients who were still sick after standard treatment. Each of these studies enrolled between 13 and 129 patients. So if you multiply however many patients per study, it's a drop in the bucket compared to the number of people that you'd need to evaluate to really get accurate assessment about ideal treatment. Above and beyond the lack of robust numbers, there were a lot of methodological flaws in these studies that I noticed that I quoted, and in some of them, the conclusions at the end of the paper, which is the part, to be honest, that doctors read, they don't read like all of the, how we did this, misrepresented the results to be more in line with the idea that short courses of treatment were adequate. The schism between the Lyme is easy to treat and Lyme is more complex factions widen with the introduction of a Lyme vaccine in 1998. So everybody, no matter what side of the debate you're on, everyone's going to recognize that Lyme disease is a public health problem and that people whose livelihood and recreation put them at risk really needed protection. So the LymeRix vaccine was put on the market, and it was actually found to be very successful in preventing Lyme disease. While the pre-marketing safety trials did not find a higher than expected incidence of long-term effects, some vaccine recipients developed arthritis symptoms after getting it. So there is a system in the United States for people to report adverse reactions to vaccines, which is called appropriately the Vaccine Adverse Events Reporting System, or VAERS. So 59 cases of arthritis were reported to VAERS, but this was out of 1.4 million Lyme vaccine doses. So that was taken as evidence by the manufacturers that the vaccine was safe, since it was such a small number and statistically no higher than the placebo group. But what about the case reports with a clear association to the vaccine? In other words, someone was completely healthy, they got the vaccine, and they developed this arthritis. So here's where we get into the difference between population-based research and personalized medicine. So population-based research is the way that we've been doing medicine for, the last of the results. So what percentage improved with the treatment? What percentage had side effects? The problem with this approach is that it buries individual reactions. So let's say that you have, 10% of people who have a bad reaction. Um, what is it about that 10% of people that made them get sick? So that's looking at things from a personalized medicine approach. So everybody has an identified or a unique genetic and environmental makeup that may make them react to interventions differently. So the defenders of the vaccine focused on the average treatment rather than subgroups to be able to say, well, what did these 59 people that got arthritis have in common? And indeed, when researchers analyzed the arthritis cases, they found that patients with a particular HLA genotype, so some genetic predisposition, produced high levels of an antibody to OspA, which is one of the antigens in the vaccine. And they create that this antibody was found in their joint fluid, and they developed chronic treatment resistant arthritis. So, how many of you have watched Ted Lasso? Okay. How many of you have seen the YouTube clip where Ted Lasso is playing darts with his arch enemy? So Ted Lasso is an American football coach who is recruited to coach a soccer team in England, and he finds himself in a pub with the bad guy of this series, and the bad guy challenges him to a game of darts, and so Ted Lasso is like, "Oh, sure, why not?" Um, and he starts to play, and he's like doing a great job. And he said, the problem with people like you is you're not curious, you're judgmental. And if you were curious, you would have said to me, Ted, do you ever play darts much? And I would have said, yes, sir, every week with my dad until I was 16. So the problem is that if you're not curious, if you don't say, that's weird, that's not an expected result, you're just going to keep on doing things the way that you've done them. And you're not going to say something like, maybe Borrelia is a more complex organism than we originally thought. Maybe because it's causing cross-reactive autoimmune arthritis, we should examine the way that it works. But they didn't. They doubled down on the vaccine safety because, after all, the statistics said that it was safe. But the vaccine was ultimately pulled off the market in 2002 because of poor sales and, by the way, a class action suit against the manufacturer. So at this point, Lyme treating clinicians figured out that they needed to band together, and they needed to work to create a counterpoint to the mainstream medicine's approach for education.
Did she freeze for you guys too?
I'm sorry.
Yep.
I think you froze for just like 2 sentences.
Oh, I'm so sorry. So what I was saying is that the International Lyme and Associated Diseases Society was created in 1999 as a counterpoint to the Infectious Disease Society of America, and their insistence that any deviation from their recommendations were not supported. So the IDSA doubled down on their interpretation of the data, and they published their guidelines on the treatment of Lyme disease in 2006 that basically did not support any of the information that was coming out from ILADS. So guidelines are pretty common Um, Different professional organizations will create guidelines. For instance, the American College of Cardiology will create guidelines for acute heart attacks, or the Infectious Disease Society will create guidelines for pneumonia. And so the guidelines for Lyme disease basically said, based on the available data and the expert opinion, here's what we feel should be done when someone has Lyme disease. And the problem with these guidelines is that they're actually kind of taken as gospel, and so someone who practices differently than what the guidelines recommend is opening themselves up to medical malpractice, to sanctions by their medical board. And so the value of these guidelines was really a problem for Lyme treating clinicians. So this was kind of the environment that I stepped into in 2006. And to be honest, before this, I kind of had similar opinions about Lyme disease as most doctors. I was aware of the controversy, but I'd been persuaded that there was something questionable about clinicians that were treating patients for years with antibiotics. And then my life changed because I received a Bush Foundation medical fellowship to study fibromyalgia and chronic fatigue syndrome. And so I had 6 months to work with different clinicians that were experts in it. And one of the people that I studied with was Dr. Karen Verkota, who recently retired from her practice in Winona. And I'll never forget what she said to me. She said, 60% of what we call chronic fatigue syndrome in Minnesota is really chronic Lyme disease. And with that simple sentence, my life changed. So I jumped into the Lyme world. But I kind of thought it was going to be temporary, because when I read the data, it really just felt like it was a matter of time before I'd be out of a job, because to me, the evidence was overwhelming that we were on the right track. So here we are. It's been 20 years and not that much has changed. So what has improved, what hasn't, and what's the future likely to hold? So the first thing I want to say is that over the past 20 years, more and more data has accumulated to support the idea that Borrelia burgdorferi infection can be persistent. In 2011, a Borrelia culture was introduced by Advanced Laboratory Services. Being able to show that Borrelia was alive and reproducing in a patient is like, it's basically like the holy grail of saying that this infection actually exists in this patient. Now, Borrelia is very slow growing. It's very hard to culture. And so Advanced Laboratory Services culture actually would incubate for up to, I think, 12 and maybe even 16 weeks before they said that the culture was negative. But unfortunately, the test was dismissed as producing false positives, and it was ultimately taken off the market after being threatened by the CDC. In 2014, ILADS published its own guidelines on Lyme disease, addressing the 3 specific questions for which there was enough research. So part of the problem is, if you don't have enough research to say definitively that something is true, then you have to rely on expert opinion, which is going to be potentially, biased in one way or the other. So the 3 questions were, are antibiotics useful for the prophylaxis of known tick bites? And they said that the single dose of doxycycline was not supported by the literature. The effectiveness of erythema migrans treatment and the role of antibiotic retreatment in patients with persistent manifestations. And those were based on the very limited literature, but of course were ignored by IDSA, which doubled down on its own 2006 guidelines and basically just republished them in 2020. So in 2015, Lyme.org, which is another advocacy organization, was frustrated by the lack of clinical trials. And so they decided to create something really novel, which is a patient-driven database called My Lyme Data. And you guys have probably heard of it. If you haven't, just Google My Lyme Data, and it's just an amazing trove of information. It's based on the responses of about 19,000 patients now. So, you know, it's not a double-blind randomized placebo-controlled trial, but with numbers like that, you're going to see patterns emerging. And they've actually been able to publish 8 studies about the data that they've collected. So, for example, they found that 75% of respondents had a co-infection, mostly Babesia and Bartonella, but also Mycoplasma, Ehrlichia, and Rocky Mountain spotted fever. It also identified why diagnosis was delayed. It asked people, why weren't you treated earlier? And in many cases, lack of clinician education and false negative tests were responsible. So this is going to identify areas where we can intervene and hopefully improve knowledge. Clinicians were also polled and asked what made it hard to treat Lyme patients. And the 2 big issues that came out were stigmatization by colleagues, and 39% of the respondents had been reported to their medical board or otherwise sanctioned. So you can kind of see where, when we're trying to recruit new clinicians to do this work, that it's really challenging work. The work itself is hard, but the environment around it is also really challenging. One of the most helpful aspects of the MyLyme data was looking at treatment responses. So 71% of patients who got well used antibiotics for over a year. So this was really helpful support for the idea that treatment really needs to be prolonged, and that a month or 2 months of antibiotics are not going to make it. In 2016, the tick-borne disease working group was created. It was a Health and Human Services Federal Advisory Committee that was authorized by the 21st Century Cures Act. This was a multidisciplinary committee made up of people on both sides of the Lyme debate, and I would have loved to have been a fly on the wall. Actually, Dr. Betty Maloney was on this board, and I can just imagine the story she has to tell about the debates. So the idea of this group was that they were going to review the research, they were going to identify gaps in treatment, and submit reports to Congress. So from 2016 to 2022, they did produce some really significant findings. So the first one I'm going to quote is, following the acute phase of infection, 29% of patients treated with recommended courses of antibiotics reported moderate to severe symptoms at 1 year, with an additional 14% reporting functional impairments. So that's pretty impressive that that was admitted. It's been difficult to ascertain the proportion of these patients accurately, and the duration of treatment could influence the outcomes. So this is a huge compromise, because 29% of people not getting better is, really kind of pokes holes in the idea that this is easy to treat and easy to cure. The second thing that they reported is chronic infection has been hypothesized to be the cause of persistent symptoms, and animal studies have demonstrated that biological cure may not be uniformly achieved with monotherapy, such as doxycycline and ceftriaxone. Now, I'm going to read this again with a little emphasis. Chronic infection has been hypothesized to be the cause of persistent symptoms, and animal studies have demonstrated that biological cure may not be uniformly achieved with monotherapy. But even with those qualifiers, this is a real milestone for it to be admitted that maybe everybody doesn't get better with the regular treatment. The third quote is, the potential benefit of extended antibiotic courses must be weighed against the risk of side effects, including adverse events of the drug and increasing antimicrobial resistance. Now, this is a huge statement. Let's talk about evidence-based medicine for a minute. Evidence-based medicine is usually thrown around as a term that means that you need double-blinded placebo-controlled trials that have to prove that something is so. But evidence-based medicine actually relies on 3 different things. One is research, another is expert opinion, and the third is actually patient preference. So the research, there's not always research to explain to us what we should do. And that's where expert opinion comes in. So expert opinion is just me saying, in my experience, when I've treated Lyme patients, this is what happens. Or an IDSA doctor saying, in my experience, when I treat Lyme patients, this is what happens. So the tiebreaker, if you don't have a lot of concrete evidence, is patient preference. Now, a patient may say, I know that I can risk getting antimicrobial resistance. I know I can risk getting C. difficile colitis if I take antibiotics for a prolonged period of time. On the other hand, my symptoms are so debilitating that to me, anything would be better than continuing the way that I am. So the fact that they are basically admitting that patient preference really does have an impact on this is actually pretty impressive. Not surprising, the IDSA did not incorporate these into their party line. So what did we do in the meantime? So in the meantime, clinicians continued doing what we could to publish. Now, let's talk a little bit about the research aspect. What do you need to create research? You need a lot of money and you need a lot of patients. The pharmaceutical industry has a lot of money, but they're not going to do a trial on doxycycline, which is, $10 for a course. And the clinicians that are treating Lyme disease, first of all, are like up to their eyeballs in treating people. The second is they don't have money to create, these massive studies that will give you the kind of data. I mean, even the conventional people were doing 13 people, 20 people. So the problem is that as much as we would like to have firm data, it's really expensive and it's really hard to create the kind of studies that we would like to have that say, yes, we have proven unequivocally that Lyme patients need more treatment than the conventional. The other challenge is the individuality of each patient. That this is not like treating blood pressure where you take 100 people and you give them all the same dose of the blood pressure medicine and you see what happens to their blood pressure. So with Lyme patients, there are co-infections. Not all Lyme patients have co-infections, but they need to be treated. People are very sensitive. So for example, Brian Fallon at Columbia University did a study where he was going to try starting people on disulfiram. Now, those of you who've been on disulfiram probably know that it's incredibly hard to take. The target dose in many cases is, let's say, 250 to 500 milligrams. When I was starting people on disulfiram, I started them on a quarter of a 250 milligram pill, so 62.5 milligrams every 3 days for 2 weeks because the half-life of disulfiram is so long. And then every other day for 2 weeks, and then every day for 2 weeks. And so you can imagine that the people that were thrown into 250 milligrams or 500 milligrams might be dropping out of the studies because they couldn't tolerate it. So just the individuality of all of these patients makes it really hard to produce the kind of data that that the conventional world looks at. But there have been a couple of case reports, case studies, 3 patients, 5 patients looking at treatment. And some of them to me are really very convincing. So for example, there was a patient of Ken Ligner's who he treated off and on for decades. And when she finally died, he was able to order an autopsy of her brain, and there were spirochetes in her brain. There was another study of, this was actually in monkeys. This is the Monica Embers trial, where she infected macaque monkeys and let them be sick for a while, then treated them for a month, and then sacrificed them, as they say, killed them, and they had active Lyme. So So there are individual bits of data that we've managed to encounter, but not the kind of robust kind of trials that we really, really need to do. So we worked on the seat of our pants, learning new strategies. So one of the most important questions on why people don't necessarily get better is why infections can persist is, again, going back to this individuality, that every person has an immune system that is affected by their genetics. Some people have a stronger constitution than others, and by their environment. So we think about infections as being, one and done. So you get a cold, our immune system jumps in and destroys it. We develop antibodies. We never get that cold again. But in reality, germs are much more persistent. So we are actually full of bacteria and viruses and maybe even parasites that our immune system has been able to keep dormant so they don't make us sick. So think about chickenpox. So if you're a kid that gets chickenpox, you obviously get over it after an uncomfortable week or two. But decades later, the chickenpox virus that has been lying dormant in your nerve roots comes out and gives you shingles. So the first thing to do is to look at why are some people able to keep infections dormant and why are some not? And I would hypothesize that there are people who get infected with Lyme disease and their immune systems just like kick its butt and make it dormant and it never bothers them for the rest of their life. There are other people who are able to, with the help of antibiotics, maybe, to do 10 days of doxycycline and they're fine. And then there are other people who don't. And some of it's genetic and some of it's environmental. And we'll talk about some of the environmental factors later on. But generally, the idea is that because infections are more tenacious, they really they can really have a hold on our bodies that we can't even say that someone's cured from Lyme disease. The best that we can say is like, well, maybe it'll stay dormant for the rest of your life. So what are these things that influence us? So if somebody just has a generally weak immune system, they're going to be at greater risk. And this can be genetic and it can also be environmental. We know that people that are sleep deprived or eat a nutrient poor diet, their immune system's not going to be as strong. We also know that genetically there are probably people who are at more risk, not because their immune system isn't strong, but because they have the Achilles heel in their immune system that allows Borrelia to evade their immune system better. So there are all kinds of reasons why people get better and don't get better, and we really need more research into that. I want to talk about a couple of other effects, specifically mold toxicity. Now, I have had more resistance from my patients about mold than anything else. That, they're willing to sleep 8 hours, they're willing not to drink 6 cans of Mountain Dew, but they are not willing to get their house tested for mold or do a mold test themselves. And I can totally understand it, but I cannot emphasize enough how, in my experience, when patients plateau or relapse or have, terrible reactions to very small amounts of treatment, that more often than not, mold is the issue. So I really want to encourage you guys, if you haven't made progress, to consider that. The other thing that's really been interesting is mast cell activation syndrome. So I'd asked earlier if people know about mast cell activation syndrome. Mast is like the mast of a ship. Mast cells are a subset of your immune cells, of your white blood cells, that are on the front lines of protecting you from infection. But if they are hyperreactive, they will tend to go off even when there isn't really a threat. So people with mast activation syndrome or MCAS tend to have really strong, what are considered allergic reactions, but can also have, gut reactions, brain reactions, any other part of their body. And so what we've learned over the years is that when people have MCAS, it's absolutely critical to treat their MCAS, to get it under control before we can start treating their Lyme, because when we try to treat the Lyme and the inflammation that ensues from that, the mast cells just go crazy. So the other thing that's been heartwarming over the years, and that I think we've made progress in, is identifying new treatments. So there is something called high throughput testing, which is an automated method that's used in drug discovery. So you can take literally thousands of drugs and test them in vitro. So it is test tube. You drop it in the test tube with the Lyme bacteria. But at least it's a good way of screening to identify what, and it's not just antibiotics, it's other things too, might have high activity against Borrelia. And probably the most famous one of these is disulfiram, which is not an antibiotic. It does have antimicrobial effects, but it is now being used to treat Lyme and Babesia very effectively. We also figured out that methylene blue works against Bartonella. We discovered the role of biofilms in persistent infection. And do you guys know about biofilms? Yeah, everybody knows about biofilms, hopefully. Yeah, they are the fallout shelters that Lyme can hide in and be protected so that even if you're on antibiotics, you don't get better. And so besides, finding these things out, the other thing that has been very robust is attempts to educate other doctors. So I mentioned Dr. Maloney before. She has actually presented at the Minnesota Academy of Family Practice several times. She and I are actually involved with Vectorwise CME that was mentioned earlier. This is an online course for people to learn about Lyme disease at their own pace. And so I do really think that we've made progress. But back to the questions that I asked before. What has changed since 2006? What hasn't? And what's the future likely to hold? So if I had given this talk a year ago, I would have said that we've made progress medically, but we're stuck politically. And I would have expressed disappointment that we're not much closer to the complete integration of our approach to Lyme disease into mainstream medicine. But I would be hopeful that eventually things would change, more due to patient advocacy and things changing and putting pressure on the medical community to make more of our position a mainstream position. Unfortunately, I think that the IDSA our opponents in general have painted themselves into a corner, and it's going to be really hard for them to admit that they're wrong. So what's changed in the last year? In May of this year, Health and Human Services Secretary Robert F. Kennedy Jr. made Lyme disease a priority of his agency. He's approved a multi-million-dollar pilot program focused on tick control, collaboration with the Cohen Foundation, which has done a lot of great work supporting Lyme research, and has pledged up to $2.5 million in innovation challenges, such as educational tools, public awareness campaigns, and a collaboration with iLads to help patients connect with experienced providers. I have to tell you, when I read the thing about iLads collaboration, I nearly fell off my seat. That was not on my bingo card. So on one hand, I am excited about the support for Lyme research and education. On the other hand, I am worried about the political fallout. Depending on the results of the midterm elections, possibly, and the 2028 presidential race, this governmental support may or may not be around for much longer. So, given the concerns about RFK Jr. within the medical community, I'm just concerned that iLad's alignment with him will probably end any possibility in the near future of reconciliation between it and the IDSA. So I, in some ways, I'm more hopeful, and in some ways, I'm a little bit more nervous. So, am I disappointed? Am I still hopeful? So, I would have really liked to have finished my career by having arrived in the promised land, so to speak, to arrived at a place where my services were no longer needed. But I will quote Martin Luther King Jr.'s famous last speech. Well, I don't know what will happen now. We've got some difficult days ahead, but it really doesn't matter to me now because I've been to the mountaintop and I've looked over and I've seen the promised land. I may not get there with you, but I want you to know tonight that we as a people, we will get to the promised land. Now, I am not Moses and I'm not Martin Luther King Jr., but I have been honored to be on this journey for 20 years, and I know that we will eventually prevail. It was not Mahatma Gandhi who's usually quoted for this. It was a trade unionist named Nicholas Klein in 1918 who said, first they ignore you, then they ridicule you, then they attack you and want to burn you, and then they build monuments to you. So I am hoping and praying that eventually the truth will prevail. And I hope that all of you find the healing of body and spirit that you seek and have lives of purpose and service. Thank you so much for letting me be part of your journey.
Thank you.
Thank you.
Ah. Let's see, there were a few questions.
Oh, good.
Okay. Well, Gina,
You want me to just add? I'll add.
yeah, since there's
So what I'm doing is a whole different thing up here in northern Minnesota. What I've had is a rife machine since '09. And I figured out that knocking down Epstein-Barr, because you were bringing up the shingles, not that's my biggest
Yeah.
player, is that's what I had going on. And boy,
Mm-hmm.
they were killing all this other stuff, but all of a sudden I realized that I was losing my mind because the nervous system was out of whack from the Epstein-Barr. So anyway, long story short, I was just curious what your thoughts are on the rife because I've had great success with that.
Yeah. Yeah. Good question. So I do actually want to say something about the Epstein-Barr virus.
Okay.
So Lyme, I think, lets other things out of jail. So you may have had Epstein-Barr virus since you were 15, but
Yep.
it's been dormant. And now Lyme is like opening all the jail cells for all of the other things to let them get out.
Yep.
Um, and you're right. I mean, you need to treat things like Epstein-Barr virus just to kind of knock down the viral load and like start getting some traction on this. And then in terms of the Rife machine, I have heard anecdotes of it being phenomenally effective. I've also heard anecdotes of it being completely useless for people. And this is the kind of thing that we really need research. Like, I would love to take, 100 people with Lyme disease and give 50 of them a Rife machine and give 50 of them like a placebo machine and see what happens. Because until we get that kind of data, we're not really going to make much progress in terms of like going beyond the anecdotes. Now, even within that kind of a situation, what if you have 5 of those 50 people have complete remission of their Lyme and 45 of them don't? Well, that doesn't mean that it didn't work. That goes back to that personalized medicine. And I would say, what do those 5 people or 10 people or 20 or whatever it is, what do they have in common? Do they have genetic things in common? Do they have the same co-infections? What makes them similar to each other so that we can now start drilling down to say your genetic makeup makes me think that you would do great with a Rife machine, and your genetic makeup makes me think it wouldn't be helpful. So
And it's dangerous, if you do it too long, it can really overkill too fast and then the
mhm,
neurotoxins. But the other thing that I've learned is that intuitively I ask questions. What is it I need to treat now? And I still do it, but I'm doing
yeah,
well, but I'm always asking every day, what is it that I need? Is there anything I need to change? But if I don't get an answer, and I trained myself to intuitively, kind of like kinesiology, but not. But
Yeah, yeah.
that's worked well because how do we ever know, what's attacking at what time? It's all so, confusing.
hmm
So I
hmm
just wondered if what you thought about all that.
Yeah, yeah, no, good observations. So one thing about the Rife machine and some of the other, I mean, like I've had patients go to Europe for a week and a half of like IV, stuff, or go to Mexico for malaria therapy and spend thousands and thousands of dollars. And I don't know what Rife machines cost now, but they're pretty expensive.
Mine
Um,
was pretty cheap, but yeah,
oh,
mine's
yeah,
a
you're
$700, but well, cheap to me, I
yeah,
guess, compared to
that's yeah.
3,000.
Yeah, exactly. Yeah. And so the more expensive something is, I think the more important it is to try to figure out. I mean, if it's a supplement that costs $30, you take it for a month and you're like, oh, I'm not any better. I'm just not going to continue it. So, but
Mhm.
I think that looking at the financial aspects of all of these treatments is important. I want to talk about the intuition. So this is an area that conventional medicine is like they're not going there. So energy medicine, I think, is really the the last frontier. Well, it's the last frontier that we know of. We'll probably find something else that's a frontier. Um, but I completely believe that people have an intuitive sense about what they need. And whether it's something like applied kinesiology or just kind of trusting your gut, it really does make sense to utilize those skills if you have them, um, and to work with your body as much as you can. So I think you're lucky that you can do that. Not everybody can, but I absolutely think that there is so much more to you know the idea that we are um, spiritual beings having a body experience rather than a human body having a spiritual experience.
Yes. And then just the last thing I had written was
Yeah.
I don't know, the Horowitz new protocol. He's talking about 8 weeks of dapsone, double dapsone is going to help bring the markers back on the Alzheimer's. I thought that was exciting when I listened to it. But what do you think about the dapsone?
Yeah. So I talked about dapsone with some of my colleagues, and I've had a couple people come to me and say, like, I really want to do this dapsone protocol. Um, it's very challenging and either the single dapsone or the double dapsone. A double dapsone is even harder on people, but um, it takes a lot of um, brain power, which sometimes really sick patients don't have, to
Yes.
figure out, like, I have to take this pill now and that pill then, and I have to go get my labs checked and stuff. Um, the experience in the people that I know is we haven't really had a tremendous amount of success. I mean, we haven't done a lot of it, but the few cases for people that I know, it has been challenging. So I'm not saying that it doesn't work, and I'm not saying that somebody shouldn't try it if they haven't done it. But I just don't have a lot of experience with it working.
Probably needing a lot of detoxification if you're doing that much because your neurotoxins are just going to be through the roof, I assume. So
Yeah,
it's tricky. You
yeah,
have to know when to do what. It's
yeah,
so
yeah, yeah.
confusing.
And I suspect that you could come up with like a modified version of it that inched people in, kind of like I was talking with the disulfiram of like maybe you start with a quarter of the dapsone dose and you go up to half of the dapsone dose and you ease into it so you
Sure.
don't have that like burst of toxins just flooding your system and making you really sick.
Yes. Sounds like a good idea.
Um,
Has anybody on the call tried the dapsone protocol?
Mm, yeah, I just listened to his thing and it sounded like there was a lot of people that he, treated that it worked on. But yeah, like you say, it's everybody's different. And if you got Epstein-Barr or you don't, or if you have, it
Yeah,
makes a
yeah,
difference.
yeah.
this is completely off the mark, but I was wondering about the biobank. How um, Do you think that is important for the future of research? And is that?
Are you talking about the Lyme Biobank where they have material from patients to like look at? Um, I do for a couple reasons. One is just to be able, and I don't know how far back the biobank goes. I think I've read that maybe um, there are samples, not necessarily in the Lyme biobank, but people have gone back and looked at like Mayo Clinic blood from the 1920s and identified Borrelia. So I do think that it is very helpful, um, and especially as we learn more and more about genetic predisposition to things, it would be interesting to take a bunch of samples and say like, these are people that weren't getting better. Do they have anything in common with each other in terms of their genetic makeup? So yeah, I think that the biobank is a great idea. The other thing that I think is, kind of a morbid thing, but can be helpful is if someone with Lyme disease dies and they donate their body, especially their brain, to be autopsied to see if they have, active Borrelia. And I mean, the ideal thing would be to actually culture it out of somebody's brain because that's incontrovertible evidence that this was not just like, oh, those are just dead bacteria that are lying around. That like this is like a living organism that can reproduce.
So, well, and that's not done just at the biobank. You could request that to be done.
You mean to no? Oh, you mean the autopsy stuff?
Yeah.
No, there are. I'm trying to remember off the top of my head. I can't remember where where you can get that kind of tissue evaluation. But I know that there are places that will do it and look for it.
I know that the U of M has a donate your body to the U of M, but I don't know if you can specifically ask for things.
Yeah.
But yeah.
Okay. I'm gonna Google this. To donate post-mortem tissue or arrange a research autopsy for Lyme disease, contact the Lyme Disease Biobank. Look at that with the National Disease Research Interchange. And it has a phone number, an 800 no matter. You have to pre-qualify and have an official diagnosis. So it's possible that, this is something that you should arrange before you actually die. Yeah. And that's Bay Area Lyme Foundation was reporting that.
Okay. Thank you. Anybody else have any? Kathy, do you have any questions?
Oh. I have a question.
Mm-hmm.
Are there other so I've been sick for 22 years and I feel like I used to be like a hearty, strong person. And now I just feel like such a delicate flower and I'm so sensitive to medications and stuff. Like, are there other infections or diseases or anything that make people so sensitive to medications and just general stuff? Or is this sort of a hallmark of tick-borne illness?
Well, I think it can happen, partly with the treatment. I mean, if you've been on antibiotics for a year, your gut is not going to respond the way that it used to, so you're going to have, more food intolerances, for example. You might be more sensitive to things. And so I don't know. I don't think of Lyme disease as necessarily like making you more sensitive, but it certainly is possible. And it's certainly possible that the other things, the changes in your body with the Lyme disease can contribute. And of course, the treatment can do it as well.
Well, in the analogy of you saying about letting other infections or issues out of the jail cells, that made sense to me too.
Yeah.
Yeah.
Yeah. Tom has a comment. Our son, age 19, was tested for Lyme in early 2024. Negative result. Many doctors meeting since for severe 24/7 headache and fatigue. we want to have more Lyme testing. So, Tom, my heart goes out to you. It's just so frustrating when you especially see your children going through things like this. And there certainly are other tests for Lyme. I mean, I mentioned the conventional tests, but I failed to mention that there are companies that are doing a much more robust kind of testing. So there's a lab called Medical Diagnostics Lab, MDL, that does a Western blot. So they don't do the ELISA. You can just order the Western blot, and it's what's called the expanded Western blot. So the CDC decided that certain bands, certain antigens were going to be in the Western blot that would be considered like positive for having Lyme, and other ones would be left out. And in particular, the 31 kilodalton band was one of the antigens in the Lymerix vaccine, which, does anybody remember when it was taken off the market?
No.
25, close to 25 years ago. So, but still, the 31 kilodalton band isn't considered in the CDC positive result. So MDL is cheap. I think it's like less than $100 if you pay out of pocket, and they actually do take some insurance. So that's a simple way to start. I failed to mention IGeneX, which has been this amazing company at the forefront of doing a ton of different work. They have their own expanded Western blot. They also do other kinds of testing. They look at they don't do culture, but they'll look for PCR. So they'll look for the genetic fingerprint in blood or in joint fluid, in spinal fluid. And they can be very effective in identifying Lyme disease. There is a Infecto lab, which is now called iLabs, which is very confusing because it sounds like iLads.
Yeah.
But Infecto lab, which is actually based in Oakdale here, do a different kind of testing called a T cell testing. So there's the B cells, which are the antibodies, and then there's the T cells. And so it's just looking at immunity from a different perspective. So there are a lot of different ways that you can test if that's what you think is going on. Sorry. Yeah, so if you suspect Lyme disease, especially if somebody has a suspicious story. So one of the things that I always tell the people that I'm teaching is, just because you have a hammer in your hand doesn't mean everything is a nail. And I think that Lyme doctors may have a tendency to have a confirmation bias where this patient's coming to be treated for Lyme disease and just assume that that's what they had. And over the course of my career, I've actually had a couple patients that I've said, like, I don't think you have Lyme disease. You know, I might be able to tell them what I think it is, but I don't think it's Lyme disease. So I do think that if somebody has a story that's really consistent, backpacking the Boundary Waters, came home and had a summer flu, had a Lyme rash, had an attached tick, the likelihood that it's Lyme disease is greater. But if you go to somebody who is competent, they're going to figure out, like, is it Lyme disease? Is it not Lyme disease? And you mentioned that your son was just diagnosed with ME/CFS, myalgic encephalitis chronic fatigue syndrome. That's really just kind of like a label. So it's like saying you have a sore throat. It doesn't tell you why you have a sore throat. ME/CFS doesn't tell you why you have fatigue. God does not smite you with ME/CFS. It comes from someplace. And so trying to drill down and find the underlying reason, secondary infections. One of the silver linings of the COVID pandemic is all of the people who have been completely incapacitated by long COVID. I'm sorry that they have it, but it is highlighting this, persistent post-infectious fatigue. And I'm sure that from the research that's being done on post-chronic fatigues or post-COVID chronic fatigue syndrome, that we're going to get some insights into post-Lyme chronic fatigue. So.
Brett, should I have Tom? Tom, do you have a Lyme doctor that your son is seeing? We kind of, Minnesota Lyme kind of has a list of the local doctors that we're happy to share with. We just don't have it like on our website.
Yeah, so you could contact Minnesota Lyme Association and get names of somebody. And like I said, most of the people that treat Lyme will also be able to say, like, is it Epstein-Barr virus? Is it mold toxicity? Is it mast cell activation syndrome? So, so it's not a thumbs up, thumbs down, is it Lyme? But hopefully you'll be able to find some, some explanation. Exposed in June 2023, National Guard, Ripley, along with federal. Oh, okay, yeah. So he does definitely have, no noticeable tick bite, but ticks on him. And one of the other people had a bullseye. By mid-August, he was experiencing flu-like symptoms and fatigue. March 2024, Mayo's infectious disease clinic testing was negative. Had 100 plus doctor's appointments. Yeah, I mean, I hate to badmouth my colleagues, but the reaction of infectious disease doctors has really been frustrating. I've heard stories of people being completely gaslighted, dismissed. I don't know what's wrong with you, but it's not Lyme disease, that kind of reaction. So I think you need a Lyme literate clinician, not a conventional clinician. So I hope you can find the help you need.
Joan, do you have any questions?
No, just thank you very, very much. I really appreciate your insights and certainly been down this road.
Yeah. I mean, I really feel like it's been a privilege to be able to do this work, and I've been so happy to be supported by Minnesota Lyme Association. And I'm actually, I'm as a retirement, what I'm telling my patients is that in lieu of any presents or whatever, to like make a donation to Minnesota Lyme Association among a couple of others. So hopefully you'll get a little bit of benefit from this. So
thank you so much.
Yeah, thank you all. And like I said, I hope everyone here can find the healing that they need and be able to, be the people that they were made to be and give the world the gifts that you were meant to give.
And if we can ever help in any way as a group, we have a lot of bodies in the group, that not everybody showed up tonight, but if you ever need any kind of help from us, don't hesitate to
Thank
ask.
you.
If you need a guinea pig to lay on the table at the end of September and say, this is what mast cell looks like, give me a call.
Oh, really? Huh? Good to know.
I'm kidding. Yeah.
Oh. So great. Well, good. Well, thank you guys so much. I hope you have a wonderful evening and bless you all.
All right.
You too. Thank you.
Bye.
Bye.